Hello, here we present a work that we published in the Journal of Microbiological Methods about the estimation of single-cell RNA numbers using RNA fluorescent tagging and flow cytometry. Progresses in microscopy and synthetic engineering fluorescent proteins now allow observing individual proteins and RNAs in live cells. Here, we focus on a method to detect and count RNAs, the MS2D GFP tagging method. Shortly for this, the synthetic RNAs have multiple binding sites for viral proteins MS2 which carry GFPs. Once these bind, the target RNAs appear as bright spots because their fluorescent intensity is much higher than the background. Interestingly, when produced, the spots are very stable, with lifetimes of several hours. However, while apparently easy, it is actually quite hard to reliably count RNAs from the images. We refer to our manuscript for a detailed description of these difficulties. Consequently, segmentation takes many working hours by specialists, even when supported by software with methods tailored for this purpose alone. This limits how many cells can be analyzed. For example, 1000 to 5000 cells can take weeks. To address this, we developed a method to count RNAs in single cells from flow cytometry data. To achieve our goal, it is necessary that when MS2 GFPs bind to the RNA and form a spot, the total cell fluorescence increases. To verify if this is true, we performed two tests. First, we tested if cells with tagged RNAs have, on average, higher fluorescence than cells without tagged RNAs. Second, we tested if, as more spots are produced over time, this total fluorescence increases. If these tests hold true, it should be possible to estimate the statistics of single-cell distributions of RNA numbers from flow cytometry data. For our tests, we use microscopy, spectrophotometry, and flow cytometry. First, for microscopy and using the cell segmentation software CellAging, we confirm that more RNAs are produced as induction strength increases. Next, knowing that induced cells produce spots over time, we compared the fluorescence intensities of cells with and without induction using a spectrophotometer. We find that cells producing spots have increasingly higher fluorescence than when not producing. Finally, we measured cells' fluorescence at different induction strengths by flow cytometry. To account for different cell sizes, we normalized the Fitch signal by the pulse width, which we denote by Fw. Note how the mean Fw increases with induction strength. Meanwhile, in cells without the target gene, the fluorescence does not increase with induction strength. We thus conclude that the increasing fluorescence is due to increasing number of tagged RNAs in the cells. We next measured by microscopy the RNA numbers produced by our gene of interest under the control of LACARA1 promoter for different concentrations of IPTG. The top figures shows the mean, standard deviation, and skewness of the single-cell distribution of RNA numbers as a function of IPTG concentration, respectively. The middle figure shows the mean, standard deviation, and skewness of the single-cell fluorescence distribution measured by flow cytometry at the same induction strength. Finally, from the bottom figures plotting mean versus mean, variance versus variance, and the third moment versus third moment of microscopy and flow cytometry data, we find that they are linearly correlated. Similarly, from flow cytometry data alone, we find that the protein statistics follows the RNA statistics. Namely, for mean, standard deviation, and skewness, the protein fluorescence follows the tagged RNA fluorescence. This confirms that the statistics of distribution of Fw value is the result of single-cell distribution of MS2D GFP tagged RNAs. To achieve our goal, it is necessary that when MS2 GFPs binds to the RNA and form a spot, the total cell fluorescence increases. To verify if this is true, we performed two tests. First, we tested if cells with tagged RNAs have, on average, higher fluorescence than cells without tagged RNAs. Second, we tested if as more spots are produced over time, this total cell fluorescence increases. If these tests hold true, it should be possible to estimate the statistics of single-cell distributions of RNA numbers from flow-cytometry data. We started by calibrating the difference between a pair of conditions from flow cytometry data, for example 0 and 1000 micromolar IPTG, to the difference between the corresponding pair of conditions from microscopy data. This process has to be done independently for the mean, variance, and third moment, but one can use any pair of conditions for each of the moments. Figure A shows the estimated mean of the single-cell distribution of RNA numbers from flow cytometry data for each condition using the calibration line obtained by using the pair of conditions 0 micromolar IPTG and 1000 micromolar IPTG. Figure B shows the estimated standard deviation of the single-cell distribution of RNA numbers from flow cytometry data for each induction level using the calibration line obtained using the pair of conditions 6.25 micromolar IPTG and 1000 micromolar IPTG. Figure C shows the estimated skewness of the single-cell distribution of RNA numbers from flow cytometry data for each induction level using the calibration line obtained using the pair of conditions 50 micromolar IPTG and 1000 micromolar IPTG. To evaluate the accuracy of the estimated mean, standard deviation, and skewness from flow cytometry data, we plotted them against the corresponding actual values obtained by microscopy. In figures D, E, and F, the blue line is the best fitting line to these points, and the black line is the ideal line if the estimated values matches exactly the actual measured values from microscopy. If the estimations are accurate, one expects the best fitting line to exhibit a 45-degree inclination and to intercept the y-axis at 0. Using the analysis of covariance test, we compared whether the best fitting line and the ideal line could be distinguished in slope and intercept in a statistical sense. Results of these tests for the mean, standard deviation, and skewness show that the best fitting line cannot be distinguished from the ideal line, from which we conclude that the estimations are good. Overall, we conclude that collecting data using microscopy from two conditions differing in RNA numbers allows accurate estimations of the mean, standard deviation, and skewness of single-cell distributions of RNA numbers from the distribution of total cell fluorescence measured by flow cytometry in multiple conditions differing in induction strength using MS2D GF tagging of RNA. The technique proposed here can support studies of gene expression dynamics as it allows using detailed microscopy data to derive conclusions and then support these conclusions with data from many cells obtained by flow cytometry. It is also interesting to note that the noise in the two measurement techniques is of different nature, implying that the combination of the two techniques should result in reliable conclusions. For more information, please read the article cited. Thank you.